So let's leave carbohydrates now and move on to fats. And just a reminder of the structure of triglycerides. It's the 3-carbon glycerol molecule on the left and the vertical, and that's esterified with 3 fatty acids, which are the lipid part of the triglyceride molecule. And triglycerides are another major source of energy and the most calorie dense. And you'll remember that both carbohydrate and protein contain 4 calories per gram, whereas fats contain 9 calories per gram. But lipids can only be oxidized to yield ATP under aerobic conditions. So there's no anaerobic options for the fatty acid part of triglycerides. And as you can see from the diagram, fats can't enter anaerobic glycolysis, so are metabolized to acetyl-CoA. And here we have another diagram to show where triglycerides feed into cellular respiration. I've left glucose in so you can see the relationship, but triglycerides, when they undergo lipolysis, so the lysing of lipids, we obviously yield glycerol and fatty acids, so separating those 2 aspects of the triglycerides, and glycerol and fatty acids are obviously the components of a triglyceride molecule. Glycerol feeds into glycolysis because it's essentially a carbohydrate-based molecule, although the fatty acids are catabolized by 2-acetyl coenzyme A and feed into the Krebs cycle. And there we go. A little text just for you to learn and read up on. So the first step of fatty acid oxidation is called beta oxidation, where the long-chain fatty acid is catabolized into 2 carbon chunks, and they're cut off one at a time, and each of these chunks are converted into acetyl coenzyme A, and that process is beta oxidation, which requires a number of B vitamins, as you can see there on the diagram in the yellow, as well as the amino acid carnitine. Or I say amino acid, it's actually a compound amino acid. I'll come on to carnitine a little bit later on. It's not a true amino acid per se. And you do get quite a bit of ATP from this process, 17 ATP, a bit better than glycolysis, which yields just 2 ATP. 1 molecule of the 18-carbon saturated fatty acid stearic acid, for example, which is obtained in both plant and animal foods, more from the latter. I think beef is quite high in stearic acid, produces 148 molecules of ATP. So I'll just put that in as a little example when it's been beta oxidized. And remember that the whole process occurs in the mitochondrion, being a solely aerobic process. So the fact that fats produce more ATP per gram than carbohydrates means they're much more energy dense. But why do you think that is? Why are they more energy dense? Or why are fats and lipids more energy dense than carbohydrates? Why do you get more ATP from fatty acids than from glucose, bearing in mind that this is an oxidative process? I wonder if you can guess. The clues to the answer is in the simple biochemical composition of fats. Can you remember what atoms they're comprised of? Well, fats, remember, are just a carbon and a hydrogen. A fatty acid, I should say, is just a carbon and a hydrogen. So there's no oxygen present. So no oxidation can occur within the molecule structure. But glucose and other carbohydrates, as you know, are carbon, hydrogen and oxygen. So glucose has already been partly oxidized, if you like. It already contains oxygen. So fatty acids have the potential, simply from their chemical structure, to undergo a greater degree of oxidation gram for gram than glucose. So it's more energy dense. And both saturated and unsaturated fats undergo beta oxidation. So this is the same with both. I said I'd mention carnitine. It's needed to carry fatty acids across the mitochondrial membrane to take part in this beta oxidation process. It's most readily available from muscle meats, hence the name carnitine, which comes from the name carn, which meaning flesh, like carnal knowledge, shall we say. But it can be synthesized in the body as well. However, synthesizing carnitine, not the most efficient process, particularly if there's a deficiency in lysine or methionine, which are needed to make carnitine. I mentioned it's often called an amino acid, but it's not a true amino acid, although it's an amino-based compound. It's synthesized from lysine and methionine. A deficiency of carnitine can lead to an inhibition of fatty acid oxidation and reduced energy production from fats. And this is quite important because a lot of our cells utilize fat as an energy source. So it's an important molecule. I mentioned lysine as well. That's an interesting amino acid. It's the antagonist of the amino acid arginine, which is abundant in a plant-based diet. Both compete with each other for the transportation both across the gut wall and also across the cell wall. So vegetarians who do not consume much animal products such as dairy or eggs may be low in lysine. Interestingly, from a health perspective, from a nutritional therapy perspective perhaps, the very common virus, the herpes virus, requires an arginine-dependent protein to replicate. Because lysine, remember, is the antagonist to arginine, additional dietary or supplemental lysine can and has been shown in the research to be beneficial in the management of herpes virus, as it reduces the use of arginine in the cell. But for those of you who get your cells or know anyone who gets cold sores on the lips and things, putting on those lip balms which contain lysine really is a bit of a waste of time. It's a bit of a gimmick, because remember, you need transporters to transport those amino acids across the cell wall, and the outer epithelial cells which line your body, so your skin, your intestines, things which line the mucous membranes and so on, which are exposed to the outer environment. It's only within the intestines that those carrier molecules, those carrier transporters are found. So they're not found on the skin around the lips. So you're not going to be taking in any lysine to do what you're intending. Bit of a gimmick, really. So transport of triglycerides. Let's have a quick reminder of how triglycerides and fatty acids are transported around the body. They're transported by lipoproteins. Triglycerides are digested and absorbed into the epithelial lining of the gut and are then repackaged up into chylomicrons. Let's have a look at those. There we go. There's a chylomicron. The chylomicron body is synthesized in the intestinal epithelium, and they carry around inside them the fatty acids, as you can see in there. So then the chylomicrons pass into the lymphatic system. Do you remember that fats are absorbed through the villi into the central lacteal, which is part of the lymphatic system, as opposed to into the blood circulation? And in the lymphatic system they circulate around and then enter the vascular system, and from there the chylomicrons can deliver triglycerides to the adipose and peripheral tissues, but they primarily end up in the liver. So you've eaten a food with any amount of fat in it, then this is exactly what's going to be happening inside you. So it's a good way to imagine that. But then, going the other direction, there are also very low density lipoproteins, so lipoproteins, so fats and proteins, which transport triglycerides from the liver to the peripheral tissues, so the adipose tissues, the fat tissues, and the muscles and the heart. And basically a lipoprotein, VLDL, very low density lipoprotein, if you remember that the blood is essentially a water-based substance, and fats are antagonistic or hydrophobic, shall we say, they need to be carried around in a hydrophilic molecule, so a non-polar molecule, and they are therefore carried around in these chylomicrons, or these lipoproteins. And that's the purpose of having those there. So let's have a look at the lipoprotein structure. There's the protein around the outside, called the apolipoprotein, then the phospholipid layer, and carried inside you've got the cholesterol and the triglyceride compounds. So what happens when chylomicrons, or the very low density lipoproteins, transfer their triglycerides to adipose tissue is that there's an enzyme on the surface of the adipocytes, so the fat cells, called lipoprotein lipase. And this hydrolyzes triglycerides, so it catabolizes them into fatty acids and glycerol. And the glycerol doesn't enter the adipocyte, but it circulates back to the liver in the blood. So the glycerol portion of the triglyceride. So you've still got the fatty acids there remaining, and they do of course enter into the adipocyte, and once in there it's enzymatically rebonded to glycerol, which is synthesized in the adipocyte to be converted back into triglycerides, which are the storage form. The reason I mention this is because when we discuss the hormonal regulation of energy metabolism, we see that hormones have different effects on enzyme function, such as lipoprotein lipase being activated by insulin. So it really forms part of the jigsaw. And we see muscles take up fatty acids and can store triglycerides or oxidize them for energy. And then the glycerol portion, as I mentioned before, transported via the blood to the liver. What's interesting about this is that our own gut microflora plays a role in the storage of fats in adipocytes, in our fat cells. And there is quite a lot of research going on at the moment on obesity and the role our intestinal microflora play. The microflora, so the gut bacteria, can increase lipoprotein lipase activity in adipocytes, so in the fat cells, to increase the storage of fats through two mechanisms. And in the first mechanism on the right hand side of this diagram, the microflora can digest the dietary fibre, or the non-digestible polysaccharides as they're also known, down at the bottom there, which, as you know, can cause flatulence. So when the bacteria absorb the resulting catabolized monosaccharides, which they've broken down, they release gases, but they also release two chemicals called carbohydrate response element binding protein, or CHREBP, as you can see, sitting on top of the liver there, and sterol regulatory element binding protein, SREBP1. You do not have to remember this, I'm just explaining you for an interesting extra. These two chemicals are carried in the bloodstream to the liver, and they induce lipogenesis, so the synthesis of fats in the liver, so lipogenesis, the creation of new fats in the liver. And these, along with the fats from the diet, leads us onto the second mechanism on the left, where the microflora are able to produce substances which suppress a naturally occurring chemical called fasting-induced adipocyte factor, shown as FIAF on the diagram, which results in an increase of lipoprotein lipase, the enzyme, and thus triglyceride storage as well. So that's how your own microflora can influence fat storage, and it's an ongoing process right now. Free fatty acids. So triglycerides are used to transport and store fats. But when we want to oxidize fats to release their energy, they're in the form of fatty acids. So if the body wants to burn additional fats, it sends a message to the adipose cells to stimulate a hormone called hormone-sensitive lipase, which splits the triglyceride in the adipocyte. So the triglycerides are broken back down again to its component parts of glycerol and fatty acids, and then both are released from the adipocyte into the blood. Now remember, glycerol is a hydrophilic molecule. There we go, both released back into the blood. So yeah, remember glycerol is a hydrophilic molecule, so it diffuses freely through the blood to the liver, this water-based blood environment. But the free fatty acids are hydrophobic, so they bind to the protein called serum albumin. There we go. And that compound is carried in the blood to the peripheral tissue, so the muscles, etc., to be taken up into the cells and used for energy. So you see that triglycerides and the very low density lipoproteins and the chylomicrons, they're not used for energy. These molecules are either carriers or storage forms for free fatty acids, which are used for energy. And when we want to use fats for energy, they're metabolized in the blood as free fatty acids, bound to serum albumin. Now the liver has some special functions as well concerning fatty acid oxidation. Remember that acetyl coenzyme A can be generated from beta oxidation of fatty acids. Remember that pathway from fats? So fats can be ultimately converted into acetyl CoA, which is at the top of the Krebs cycle. And instead of passing through into the Krebs cycle, the liver can convert a proportion of the CoA into biochemicals called ketone bodies. And we'll come across those a little bit later on. In fact, we're going to come across them right now. These small compounds, which can then be exported from the liver, these ketone bodies, into the systemic circulation and used by other cells and tissues to break down and yield energy, they're basically metabolized back into acetyl CoA and fed into the Krebs cycle of the cells that need the energy. So they're basically just another substrate which can provide energy to the cell. And the significance of ketone bodies will become clearer later on, but this is just an introduction to these little 4-carbon organic acids. You can see the 4 carbon molecules up at the top there. The more beta oxidation of fatty acids that's occurring in the liver, the more ketone bodies will be produced. And the diagram shows us at the top beta hydroxybutyrate, and then the other one is acetoacetate, and they're interconvertible, so they can be metabolized from one back to the other. And this diagram shows how the ketone bodies are then converted back into acetyl CoA in our peripheral tissues, and the acetyl CoA can then be fed into the Krebs cycle. And obviously in the liver, when the ketone bodies are synthesized, it's the reverse process. So 2 acetyl CoA molecules are combined to form acetoacetyl CoA, and then you can see that the coenzyme A is removed to form acetoacetate, and then that can be interconverted to form hydroxybutyrate. If acetoacetate is in a high concentration in the blood, it'll spontaneously decarboxylate to form a compound called acetone. And you may have heard of acetone before. It's that compound that can be excreted out of the body through the lungs and has that familiar pear drops fragrance, like pear drop sweets. And this is a feature of uncontrolled diabetes. You may have heard about this before. So this acetone smell is not really something you want to be experiencing. a lot of ketones are produced in diabetes, so their ketone body concentration rises in the blood and you get this acetone smell on the breath. And the physiological state of having elevated levels of ketone bodies in the blood is called ketosis. And ketosis also occurs in low carbohydrate diets, such as Atkins diet and so on. And that concludes our look at fat metabolism. So let's now have a look at protein and amino acid metabolism. Very good. Now, you'll remember that the primary function of protein is to synthesize structural tissues and functional compounds such as enzymes, so their primary use is not to yield energy. Partly because amino acids contain nitrogen, unlike fats and carbohydrates, and the body can't oxidize nitrogen, so it has to be removed before it can use amino acids for energy. So it's not as clean a process as using carbohydrates or fats as a source of energy. Glucose is the cleanest, and then fatty acids are also good. However, if you have an excess of amino acids in the body, they have to be excreted because we can't store them in the same way as carbohydrates or fatty acids. And don't get to thinking that muscle is a protein store, because it's not. But we need to remove the nitrogen, and then the remaining part of it can be used in carbohydrate metabolism. So we can use it in carbohydrate metabolism, or if the body is in a state of fasting or starvation and we don't have enough carbohydrate or fatty acids even, we can start to break down tissues which comprise a high level of protein. So this would be muscles, to be used to generate energy. And though this is far from an ideal situation, really, to be in, because muscle wastage just losing muscle tissue as a result of this process is associated with a whole host of different diseases and health conditions. So it's not something we really want to be happening. So the first step, if we're going to be using amino acids for energy, is to remove the nitrogen, as I said. So that would be the amino group. There we go. And usually the first step is to transfer the amino group to a keto acid and then replace the amino group with a keto group. So what you end up with is a keto acid instead of an amino acid. And you've just found out keto acids can then be fed into carbohydrate metabolism. You can see from the diagram. Now the thing here is that we've looked at glucose and fatty acid oxidation, and in those cases you're always talking about the same compound being oxidized. So for example, carbohydrate metabolism is glucose or possibly fructose, but it still enters the same pathway in terms of pyruvate to acetyl-CoA to the Krebs cycle and so on. And with fatty acids again, you just ping off the two carbon fragments and it's the same process. But with amino acids, there are 20 different ones, and with quite different structures, as you can see from the diagram there. Because remember, you've got that different side chain, which can be as simple as a hydrogen or a methyl group, or as complex as a phenolic ring or a sulfur-containing group. So comparatively, it's a big and diverse family, and therefore the catabolism of amino acids is a lot more varied and a bit more complex than, say, carbohydrate. But the important thing to remember is that we remove the amino group, which is usually transferred to alpha-ketoglutarate to form glutamic acid, and then the remaining part of the amino acid, which is now a keto acid, so look back there again, it either feeds directly into carbohydrate metabolism or it's modified so that it can feed into carbohydrate metabolism. Okay, so let's take a step back and look at the big picture. We've already looked at carbohydrate and fatty acid metabolism, but with proteins you have what's called the amino acid pool. over on the left-hand side there, which is simply the collection of these free amino acids floating around the blood and in tissues. Remember that amino acids, just like everything else in your body, is continuously being turned over all the time. Then the ammonia is fed through the urea cycle, and what's left of the keto acid, you see the urea cycle there? What's left of the keto acid either feeds into the Krebs cycle, or it may be converted into acetyl-CoA, or it may be converted to a ketone body, or it may be converted to pyruvate. And this depends on the individual amino acid and its structure, which I just mentioned before, which essentially depends on its unique feature, which I'm sure you know is its side chain. So this structure determines its suitability for which metabolic processes it will head off to. So we've got the ketogenic amino acids or the glucogenic amino acids. But glucogenic amino acids are very important because, as we'll see later, if the body is starved of carbohydrate, it can catabolize amino acids to synthesize glucose, as I'm sure you can guess from the name. But you can only synthesize glucose from glucogenic amino acids, not from ketogenic amino acids. And this synthesizing of glucose from glucogenic amino acids is called gluconeogenesis. So we know that as well as carbohydrate, both protein and fats can be used as a source of energy. And derivatives of proteins can end up in the Krebs cycle, just like those of carbohydrates. And it really is worth familiarizing yourself with components of the Krebs cycle, like alpha-ketoglutarate in the bottom right hand of the cycle, or succinate or fumarate, malate, etc. on the left of the cycle. And some of them are present in food, so malic acid is in apples, so the acidic version of the alkaline malate in the diagram. Fumaric acid is a common food additive. Citrate, citric acid, is of course found in citrus fruit, and in functional testing that you'll learn about later on in the second year, I think, and that nutritional therapists perform on their clients, this functional testing, the presence or absence of many of these metabolites give us an indication into things such as the efficiency of the mitochondria in synthesizing adenosine triphosphate, and the presence of bacterial overgrowth as well. So they're quite a good indicator of different things which are going inside the body, be it with regards to energy production or your body's microflora situation. Incidentally, the laboratory evaluations book, because that deals with testing, functional testing, it discusses these different things quite a bit, and these are all called organic acids because they have these carbon compounds. So organic acid testing is something you can look up if you've bought that book, or I'm sure you'll find it somewhere on the internet. But getting back to this, for oxidizing amino acids to use as a source of energy, we take the nitrogen-based amino group off the amino acid, and what you're left with is a keto acid. And most of these amino acid derivatives are keto acids, so alpha-ketoglutarate, oxaloacetate. And this is where the amino groups come into the cellular respiration story. So for example, if you just swap the keto group for an amino group of alpha-ketoglutarate, you get glutamic acid, one of our amino acids. And the same for oxaloacetate. It's very simply converted to aspartate, another amino acid. And pyruvate is essentially the keto form of alanine, another amino acid. So it all links up very nicely, and there's a definite logic to all that's going on here in the bigger picture. I previously mentioned deamination, so let's take a look at what that's all about. So as its name suggests, deamination is the removal of the amino group from an amino acid, and this process normally takes place in the metabolic factory that's the liver. Usually the amino acid undergoes a transamination reaction. Remember that trans means across, like transatlantic, to cross the Atlantic. So it loses its amino group and passes that across to alpha-ketoglutarate, you can see from the diagram there on the right, to form glutamic acid or glutamate, which is its base form. And then glutamic acid or glutamate uses vitamin B3 and the enzyme glutamate dehydrogenase, and the amino group is removed from glutamate and converts back to alpha-ketoglutarate, which is then free to receive the amino group from another amino acid. And this process produces the toxic product ammonia, NH3, which is why this is not the cleanest of metabolic processes. So ammonia is the byproduct of deamination, and this process is going on in almost all cells inside you at this moment. But in order to transport ammonia to a place where it can be excreted, it's usually metabolized into glutamine, or in the case of muscle it can be used to add to pyruvate to generate the amino acid alanine, and in that form it's transported in the blood back to the liver, where we undergo the urea cycle, which is where we generate urea, which is the main nitrogen containing substance of urine. It's a reversible process, and as I said, muscles export ammonia as alanine. This part of the lecture is where metabolism gets a bit more dynamic. We've looked at the pathways and different catabolic and anabolic reactions, and now we're going to look at how the pathways which utilized carbohydrates, so the fatty acids, the proteins, are integrated in the body to make sure that at any moment in time the body can adapt its utilization of these macronutrients to make sure we have a permanent and continuous supply of energy. As I'm sure you know, or at least can guess, without this permanent or continuous supply of energy, you'd be in a bit of a pickle, and you wouldn't be lasting a second longer. So then, true or false? The body uses glucose for fuel and then switches to fats when glucose is used up. Is that true, or is it false? And I put this in to highlight that when you first start to understand about carbohydrate and fatty acids and their use for energy, and the fact that the body's preferred source of energy is glucose, there's this idea that the body uses glucose for fuel, and when it runs out of glucose it switches to using fats because fatty acids are the secondary preference. But the answer to the question is false, and the body will generally use a combination of both glucose and fatty acids at all times, provided that they are available. And we tend to think of one of the most glucose-hungry tissues as muscle, but in particular when muscles are at rest or doing very light work, they will be principally using fatty acids, and it's not until they're doing a significant amount of work that they'll start to use glucose and break down their stored glycogen supply for energy. So there we go. Right then, so first of all we're going to consider the state when our body is taking in nutrients. So this is a dynamic process. So it's digesting and absorbing food, the macromolecules. Essentially a new supply of energy is becoming available from the intestines and introduced into the circulation. What does the body do at this point in time to sort out these nutrients? Which does it use to provide energy? Which does it store up? And these are questions which you may have asked yourself at some point. And we call this the fed state, which typically lasts up to 4 hours following ingesting a normal sized meal containing a reasonable balance of carbohydrates and fats and proteins. But just like everything else, there will be a variation in the duration of the fed state, depending on the biochemical individuality of the person, which we learned that term in the functional medicine lecture, and their current metabolic rate as well as the nutrient composition of the meal. You're probably familiar with the concept that when you eat a meal that contains a significant amount of carbohydrate, your blood glucose level will rise, and the pancreas will sense the rise in blood glucose and it will release insulin, which is a hormone. Now, insulin is an interesting hormone, and like many hormones, it has a number of effects on various tissues, which helps the body to utilize the nutrients which are being absorbed. The obvious one, of course, is that insulin in the blood stimulates the entry of glucose into insulin-sensitive cells and tissues, so i.e. those which contain the GLUT4 transporters, notably muscle and adipose tissue, and a large amount of glucose is also taken up by the liver. Remember that insulin is an anabolic hormone. When we have an abundance of nutrients, we have an opportunity to build up or anabolize new molecules. So when we're in an absorptive state, when we're eating food, we have an abundance of macronutrients, then we have this chance to both build up stores of these nutrients, which in the case of carbohydrate is called glycogen synthesis, and synthesize new molecules and create new energy, which in turn allow our body to continue to function by way of the myriad of the various biochemical molecules required, which for carbohydrate is the effects of glucose uptake, which is why we eat, basically, amongst other things. So perhaps obviously in stimulating glycogen synthesis, it inhibits the catabolism of glycogen, because what's the point of releasing glycogen if you are taking in fresh glucose here? So insulin inhibits the catabolism of glycogen, as well as inhibiting the processes of gluconeogenesis, which, as you'll remember, is the synthesis of new glucose from glucogenic amino acids we were just talking about. Because clearly if you're absorbing new glucose from a meal and able to store some of it away as glycogen, then you don't need to be synthesizing more glucose by way of the less efficient gluconeogenesis. So I hope that's clear. Okay, so we know that insulin has its role to play in glucose metabolism, but it also has an important role in lipid or fat and protein metabolism. In terms of lipids, first of all, it reduces free fatty acids in the blood by inhibiting hormone-sensitive lipase, which is why I mentioned this enzyme earlier. Remember that hormone-sensitive lipase is the enzyme which breaks down triglycerides in adipocytes. It's responsible for lipolysis. Remember that? So insulin inhibits lipolysis and therefore the mobilization of fatty acids. In other words, it doesn't help fat burning. If you are not mobilizing fatty acids, then you're not going to be using fatty acids up as energy. If you're not releasing fatty acids from the cell, from the adipocyte, from the fat cells, then you're not going to be able to use them in other cells as energy. So insulin is blocking the release of fatty acids. Remember, insulin is anabolic hormone, and breaking down fatty acids for energy and so on is a catabolic process. So it's quite common sense, really. On the other hand, insulin increases concentrations and activity of the enzyme lipoprotein lipase in adipose tissue. And this means fatty acid uptake in adipocytes is enhanced from our old friends very low density lipoprotein and chylomicrons. So if you have quite a lot of fat in your meal, which has been absorbed and packaged up in chylomicrons in the circulation, if you have sufficient insulin floating around, it will promote the transfer of some of those triglycerides from the chylomicrons into the adipose tissue. So it's causing fat storage. So increased lipoprotein lipase activity, important in insulin resistance, which is a characteristic, as you know, of type 2 diabetes, where we become resistant to the effects of insulin. And what happens is lipoprotein lipase activity is not activated in the same way as it is in normal insulin sensitivity. So your circulating triglycerides don't get transferred into the adipocytes. So you get increased circulating very low density lipoprotein, increased circulating chylomicrons, which means of course raised lipid levels, so raised levels of fat in the blood, which is one of the risk factors for cardiovascular disease. And that's one of the reasons type 2 diabetes increases your risk of cardiovascular disease, because you're not getting sufficient lipoprotein lipase activity to get your triglycerides stored away. Instead, they just continuously circulate around in the blood. So if you can see that to understand metabolism is very important when you come to understand and study disease processes. The key is, it really is not to be afraid of metabolism, because I know this can be quite overwhelming. Don't be afraid of it, just sort of go through it, and it really is a logical process. There'll be lots of these, very satisfying aha moments when you come to understand how things fit together. So take a situation that applies to many healthy people. When you increase your intake of alcohol, for example, it affects lipid metabolism in the liver, which again leads to increased blood lipids. And the message I'm just trying to get across is that there are many things that affect lipid metabolism in the body. They're all normal processes, or logical processes I should say, whether they're occurring in a healthy individual or an unhealthy individual. But they're quite straightforward. We're all aware that if you eat too much sugar, it turns to fat, but the metabolic pathway for this is simply that in the liver and adipocytes, glucose is broken down to pyruvate by glycolysis. Then you have the link reaction. That then enters the mitochondrion, so the link reaction metabolizes pyruvate to acetyl-CoA, and instead of entering the already saturated Krebs cycle, the acetyl-CoA is converted into fatty acids, which uses various B vitamins, and at the same time glycerol is synthesized from glycolysis intermediates, and the two combine to form triglycerides. So it's pretty straightforward. So when you think about sugar turning to fat, it's not as simple as sugar just magically is transformed into fat at the stroke of midnight, although it's pretty simple to remember that the glucose is catabolized to acetyl-CoA, and the products along the way go to form the lipid triglycerides. In the liver, those triglycerides are packaged up in the very low density lipoproteins, the VLDLs, but in the adipocyte the triglycerides are simply stored in the cells as normal. Now insulin also stimulates the cholesterol production in the liver, which would then also be transported out in the VLDL. As well as the liver, the adrenal cortex, the intestinal epithelial cells, I think, and the reproductive tissues. Yeah, actually, as we've got on the slide there, they're the main producers of cholesterol, but all cells have the ability to synthesize cholesterol because, of course, it's an essential component of the cell membrane, as you see in the diagram in the bottom left-hand corner. Cholesterol is synthesized from acetyl-CoA, so I'm sure you can see that this molecule of acetyl-CoA is proving to be very diverse and a real multitasker. Cholesterol is also the starting point for the synthesis of steroid hormones and coenzyme Q10, which I'm sure you remember is that component of the electron transport chain. And it's the synthesis of cholesterol that is inhibited by statins, which are drugs which are prescribed for people who have been deemed as having an elevated cholesterol level. I did want to talk a bit about cholesterol here, because the biochemical actions of which of cholesterol, they're quite controversial, and a lot of what has been discussed in medicine about cholesterol is frankly spurious and demonstrates a distinct lack of understanding about cholesterol, particularly in the mass media and so on. As you know, one of its principal roles is as an essential component of the cell membrane, but it's also found travelling around in the blood, and it's this that gives rise to the hysteria about cholesterol. It's been blamed for the principal factor in heart disease, yet 50% of people who have heart attacks have what are deemed as normal cholesterol levels. But that's not to say that cholesterol is not of significance in heart disease. It is, but there are many other factors as well which need to be considered. Of course, as is the way of the world at the moment, much of the hysteria about cholesterol has been whipped up by the pharmaceutical companies that are selling the statins that claim to lower blood cholesterol concentrations. But cholesterol remains an interesting group of molecules, so let's talk about particle size first. It may surprise you that in my nutritional therapy practice, I really don't look at people's total cholesterol counts or their LDL counts. And this slide will be going to look at low-density lipoproteins. We tend to think about, we know there are very low-density lipoproteins, then low-density lipoproteins, and high-density lipoproteins, so the HDL. And some of you may also think that the ratio between the LDL and the HDL is what of significance. It's interesting to note that there are major international annual lipid conferences where the top lipidologists go to discuss advances in the science. And these conferences typically will have a group meeting to decide by voting whether we should eliminate LDL from our nomenclature, our labelling and our concern. And in talking about people's risk factors, because most lipidologists will tell you that LDL has no meaning because there aren't one but 7 different particles of LDL that are at least known now. And they are generously described as either big and fluffy or hard and dense. And I explain to my clients that LDL carries fat around the body, so those big and fluffy fellows are like removal vans which carry fat, and if you have those big professional moving company vans, these are good for you. They carry fat around safely and they deposit it safely. So there's some fat coming in and being carried around safely. On the other hand, these little hard particles are what you could call the little open top vans. These are the fellows who put the 3 rooms of furniture on the back with bungee cords, and you have your mattresses flying off down the motorway. And the analogy is quite a good one because the little guys are the vulnerable ones that get activated, they get oxidized. They're the ones that burrow into the lining of your arteries and cause the furring up of the arteries. That's a big problem in heart disease. So it's the little LDLs that we need to worry about. Well, in general, triglycerides are a very good indication of what kind of particle size you're going to make. And I use the simple example that triglycerides are the first form of fat we manufacture from sugars, as we've already discussed, and starches. And the only time in our evolution we saw an abundance of sugars and starches was when fruit ripened on a tree in the autumn and we needed to store fat for winter. And it's important to understand about our evolutionary biology, because this gives us very clear clues as to how we work and why these different biochemical processes are functioning in the way they do. So it turns out that if we're making lots of triglycerides, we overwhelm this removal van system and we tend to make quite a few of the little open top vans, like we see at the bottom there. On the other hand, if we don't make many triglycerides, we don't overwhelm any of these systems and we don't make many of these little fellows. And it's simplistic, but it's amazing how accurate this is. So the sugar and the fast releasing carbohydrates, which we are, being told to avoid and so on, really are a problem in this. In addition to the low density lipoprotein cholesterol, we have the HDL, the high density lipoprotein cholesterol, the so-called good cholesterol, which is actually 5 different particles. And there are little ones and big ones. And you might rightly guess that the big ones are good for you. So the big one, which is called HDL2B, is nicknamed Pac-Man because it literally goes around and gobbles up fat off the blood vessel walls wherever it can find it. And it's produced by the liver and goes around like an empty sack. And as it goes around, it literally fills up. And you can watch these particles under the microscope fill up with lipids, and it's quite exciting to watch, really. I've seen it in the laboratory at university. So if it's winter time, you need to go and pick up fat to use for energy. So you make a lot of HDL in the winter when you need it. And if you're trying to store fat for the winter, if you're at a fruit tree, the last thing you want to make is HDL. Because if you're trying to store fat, you don't want to be going around picking it up and scraping it off. So the key thing here are the levels of triglycerides. And this might surprise some of you who may have read the old literature about cholesterol, because triglycerides are not a prominent feature of these large population cardiovascular disease studies. But I think if I was going to only request one blood test for conditions such as cardiovascular disease, then it's the triglycerides is what I'd do. The lower the triglycerides, the better. Most people will soon learn that the ratio of HDL to triglycerides is the best predictor of avoidance of heart disease. That ratio should be at least 1 to 1, and the higher the ratio of HDL to triglycerides, the better off you are. And we have so many people walking around with what are deemed as normal levels of triglycerides and normal levels of HDL actually have a terrible HDL to triglyceride ratio. I think that's the elephant in the room as to why we see so much cardiovascular disease in so-called healthy people. So the more an individual's triglycerides go down, the healthier they get. As we said earlier, insulin is an anabolic hormone stimulating the synthesis of the new larger molecules, so perhaps unsurprisingly it also stimulates protein synthesis. In order to achieve that, it stimulates the uptake of amino acids into muscles, and like stored triglycerides, it inhibits the breakdown of protein as well, which is why athletes and bodybuilders try to eat protein regularly, or just eat generally regularly, the common 6 meals a day thing, to try and keep their insulin levels and its anabolic effect elevated throughout the day. So when we're digesting and absorbing the macronutrients from a meal, we get insulin release with some carbohydrate, stimulated by the rise in blood glucose, and that has a number of anabolic effects on carbohydrate, fat and protein metabolism, in which excesses that are not used up for energy are stored away as triglycerides and proteins, and of course glucose is the primary fuel used up at that time. I will say that in the sports literature, for those people who are trying to put on a bit more muscle, a bit more lean tissue, which is always going to be the healthy way to go about your body composition, or to develop your body composition, then taking protein and carbohydrate together has shown to be beneficial, and that's because of this anabolic effect of insulin. We know that the proteins contain the amino acids which are the basic building blocks for muscle, but it's the carbohydrate aspect of the meal as well which is going to stimulate this anabolic effect of the insulin. So protein and carbohydrate together is always preferable to pure protein on its own. At a time when there's an abundance of amino acids, there's a lot of protein synthesis going on, which is the theory why athletes consume protein on a regular basis throughout the day. And non-essential amino acids can be synthesized to form essential amino acids, and also from carbohydrate intermediates as well. So pyruvate is converted to alanine, oxaloacetate is metabolized to aspartate, and there are a lot of complex ones which involve a variety of micronutrients as well, such as methyl tetrahydrofolate from folic acid, and that's involved in the synthesis of a number of amino acids like serine from glycine. So, sounds a bit complex, but actually it's not that complex. And under the influence of insulin, a number of nitrogenous compounds can be synthesized as well. So these protein-based compounds, not just proteins but also purines and pyrimidines for constructing DNA and RNA. Obviously, if you're synthesizing proteins, you need a lot of RNA, which, as you know, is involved in protein synthesis. Remember that from your previous studies on how to synthesize proteins from DNA, RNA, transcribing and translating the genetic code from DNA into proteins. And if you're synthesizing new tissues and cells, which is constantly occurring inside your body, you need new DNA. But you also need glycoproteins, then proteoglycans for connective tissue, porphyrins such as heme, and other compounds such as neurotransmitters in your brain, creatine as well. So a large, diverse range of compounds can be synthesized from amino acids. So in the 4 to 6 hours, you can see the timeline along the bottom of this graph. The 4 to 6 hours from when you eat a meal, you get a rise in blood glucose, which is the green line in the middle of the chart, which results in a similar rise in insulin, the dark blue line at the top, and also a corresponding increase in glycogen stores, the light blue line, being synthesized in the liver. So we're storing away glycogen. Whereas the mobilization of fatty acids in the adipose tissue is not stimulated, that purple line. And there's also minimal ketone production in the liver during this fed state. So after we've eaten a meal, of those first 4, 5, 6 hours after a meal. Right then. Now, I appreciate that we have discussed a lot of things so far, so what you may like to do now, and I'd recommend you do this, is we're going to stop the recording and for a few minutes just reflect on what you've learned here. Just go back through the slides, consider the functions of insulin and possibly draw a diagram or bullet points and notes so that you can get a clearer picture for you of what the body does with nutrients in the fed state. So let's have a go at that, and then we'll come back in a minute when you're a bit clearer on what we've just been doing.